Welcome to the last lecture of Biochemistry 1. Today we're going to look at enzyme regulation. Enzyme regulation can be either reactive or passive, and passive simply means that the enzyme activity is able to respond to concentration of substrate. So you've seen before the curve of substrate concentration against initial velocity, and as the substrate concentration increases, so the reaction rate of the enzyme increases. And what that means is if there's an excess of substrate, then the speed of the reaction is going to be much faster than if there's very little substrate. And this gives a passive means for controlling how quickly the reaction proceeds. So if we look at the case where the substrate concentration is around about the range of the Km value of the enzyme, here it shows the unit change in substrate concentration and how much the reaction rate is increased by that unit change in substrate concentration. If we go further along this curve, however, and we take another unit increase in substrate concentration, we see that the increase in reaction rate is very much smaller. So for the same unit increase in substrate concentration, we're getting a much smaller increase in reaction rate at a higher substrate concentration. So for the enzyme having a Km value, which is around about the concentration of the substrates in the cell, means that the rate of this reaction can respond very well to either an increase or a decrease in substrate concentration. Biochemistry, reactions proceed generally according to a pathway, A to B to C to D to E to F. And if we consider a reaction where the delta G value is less than 0, is negative for A to F, then as a whole, this reaction is going to proceed from A to F, even if some of these reactions actually are able to go in the reverse direction. By definition, all these chemical reactions are able to go in the reverse direction, but the single forward arrow denotes that it's much more energetically favorable to go from A to B. If you have these A and B equilibrium conditions, then at standard conditions, and then leave them to reach equilibrium, then you'll have a great preponderance, much more of B than you have of A. So everything is reversible, but the forward arrow indicates that the shift is much further towards B. And it doesn't matter too much what these individual reactions are like in terms of their thermodynamic favorability. The important thing is that once connected in a system, it's the overall change in energy, or free energy, from the starting reactant to the final product. So coupling these reactions together enables us to make some thermodynamically unfavorable reactions to occur very easily and even very rapidly. And this is analogous to the flow of water in a system of pipes. And I've modified the image in your book here to show more conditions. Here we have, if we consider the high potential energy and a low potential energy water, then water is going to flow through this system from the upper reservoir to the lower reservoir. And if we consider this as an analogy to the thermodynamics of a metabolic pathway, then in this analogy, the overall delta G value is going to be negative. It's going to be less than 0, going from high to low. However, when we look at various points along the pathway, then we can notice some interesting features. First of all, here is what we can call a bottleneck. It's a rate limiting reaction, or a rate limiting step. In the case of our water analogy, water is going to flow through here, but it's going to flow through slowly. If we didn't have this, then it would take much less time for this reservoir to empty and this reservoir to fill. That's kinetics. However, regardless of how fast or slow this is going to happen, um, it's um, it's going to happen all the same. And we can control how fast or slow by this bottleneck here. And this is true also of enzymes, because the enzymes all have their own unique Km and Vmax properties, and some enzyme catalyzed steps in the pathway, or may be rate limiting like this. And these are usually the targets for controlling and regulating the overall flow of metabolites from initial reactants to the final products of that pathway. Enzymes which catalyze a rate limiting state are also very good targets for seeking new drugs. So here we have a rate limiting step or a bottleneck. Here we have a case where the delta G value, do you think it'll be 0 or positive or negative at this point in the flow of water? And then what do you think about this one here? So overall the reaction is negative, but here we're going uphill. And so delta G here is going to be 0, and delta G here is actually going to be positive. So the flow of water is going against gravity in this case, just as the flow or flux through a metabolic pathway is proceeding from the beginning to the end of the pathway, even though even though a step within that pathway may actually be thermodynamically unfavorable. So this diagram puts together some thermodynamic concepts and kinetic concept together. It's a very useful analogy. Another factor for control is the physical separation of metabolic pathways from each other. And anabolic and catabolic pathways are generally separated physically from each other. In addition, different components of pathways are separated from each other. So in terms of catabolic, which means breaking things down to produce energy, we have glycolysis and we have the citric acid cycle. And glycolysis is occurring in the cytosol, and the citric acid cycle is occurring in the mitochondria. And this separation of pathways enables much better regulation and efficiency overall. Even within the mitochondria itself, we may consider the electron transport chain and the flow of protons from the matrix to the intermembrane space. And this is the whole basis for providing the potential energy in order to drive ATP formation. So the physical separation of individual steps or sequences of steps from other parts of the whole picture are fundamental to the correct functioning of our cells. Some other examples here from fatty acid biosynthesis in the cytosol and oxidation in the mitochondria as well. We can also regulate how much enzyme we have, and it makes sense that we'd want to be able to do that. It turns out that for all proteins, and enzymes are for the most part proteins, then there is an equilibrium between protein or enzyme and amino acids. So enzymes are made up of amino acids, so there's a rate for the synthesis of an enzyme, but also enzymes get degraded along with every other protein in a cell. And for any given enzyme or protein, and here is an example table for various different proteins, looking at how long they survive in our cells, or what's the actual half-life, which means how long it takes for the level of this protein to drop to a half after synthesis. So for this one, HIF1 alpha, we've got 50 minutes. For I kappa B alpha, 100 minutes. For P53, a vital tumor suppressor protein, just 20 minutes. NRF2, 10 to 15 minutes, and 300 minutes for this one. So for any given protein, there's a normal half-life for that protein. It doesn't live forever. And it's an amazing turnover. A lot of energy is expended in the cell, constantly remaking things. So these are broken down within minutes, I mean, many minutes, 50 minutes, 100 minutes, but they're broken down. And if the cell still needs these, they have to be made again. And this can be controlled. Here is an example of various inducers which are able to change the half-life of these proteins. So for HIF1 alpha, which is a hypoxia inducible factor, then the presence of hypoxia, which is low oxygen pressure, partial pressure, actually greatly increases the half-life of this factor. So it's an unstable factor, but when the oxygen pressure is low, then this factor is needed, and it remains in the cell for 4 or 5 times longer compared to the normal state. In this example, the opposite is the case. So certain TNF alpha and other inflammatory cytokines, they can cause a great reduction in the half-life of this protein, which favors increased transcription of various proteins or various genes which will code for proteins that are normally inhibited by this factor that prevent a transcription factor from entering the nucleus. So, and likewise other examples along here. So let's look briefly at what is controlling the synthesis and the degradation of proteins. So synthesis is maybe constitutive, but even if it's constitutive, everything still has its own half-life because there is degradation at the same time. But synthesis can be altered by extracellular signals, cytokines, hormones, and such as, say, estrogen or testosterone that can pass through the plasma cell membrane and can then, or a part of it can then become a nuclear receptor or combine to a nuclear receptor, which will translocate to the nucleus and stimulate gene expression. Gene expression control could be active, so we can increase the gene expression, or it can be negative and we can switch off gene expression. Also, our nutrients, so what we eat is actually affecting our metabolic state, and that can drive through a series of steps changes in things which control the acetylation of histones, which are wrapped together with our DNA as chromatin, and this is known to be a vital control mechanism for gene expression. So by modulating the architecture of chromatin, then we can alter gene expression. And various metabolic enzymes have actually been found to even be able to enter the nucleus, and they have some dual functions in the cytosol and in the nucleus, and some of them can affect gene expression as well. So the synthesis of enzymes can be controlled. We can increase or decrease that. In addition, the degradation is under control as well. In order for proteins to be degraded, they have to be told that their time is up, their number has come. Intahat al-ajal in Arabic we say. And this occurs through a series of other enzymes, actually, ubiquitin-relating enzymes, which add a small protein called ubiquitin in a chain onto target proteins. And this acts as a signal telling the cell to direct this protein towards a large tryptic body called the proteasome, which will break up this protein into lots of small pieces. And those small pieces, they can be amino acids or peptides, and peptides are actually expressed on the surface of the cell, and we call this antigen presentation. This presentation occurs via complexing of these peptides to MHC molecules. And this gives the immune system a way to actually see what's inside the cell, because if these are our normal metabolic enzymes being turned over regularly, then our immune system ought not to see those, because the immune system has been trained to not react against itself. But if these proteins come from an infecting virus, then the immune system will recognize these once they're expressed on the surface of the cell, and will begin mounting an adaptive immune response via T lymphocytes. So we have two ways to control. We can control the degradation, and we can control, or the cell can control, the rate of synthesis. And multiple factors can affect both synthesis and degradation at the same time. However, controlling enzyme quantity by controlling the rate of synthesis or the rate of degradation is slow, and the effect is felt over a period of hours. In order to have much faster responses, then the um, the catalytic efficiency of the enzymes themselves can be changed. And this can be done via dissociable ligands, small molecules that can either inhibit the activity of any of these enzymes, or which may actually increase the activity of these enzymes. And what we have here is an example of a pathway with some branches on it, towards, from various intermediary substrates towards some final end products, A, B, C, and D. And in this case, a red arrow has been drawn back to the first step unique to that pathway. So if we want D, then we need to have S substrate number 3, and substrate number 3 could be directed down any of these pathways. In order to control the pathway that makes D, and in order to switch this pathway off, if we have enough D, and as the concentration of D increases, then one mechanism which is quite common in our cellular metabolism is what we call feedback inhibition, which is also called negative inhibition, where the product limits its own production, not by the step directly preceding it, but by um, the first unique step going towards that product. So that's this step here between S3 and S5 for D. For B, for example, then here is the first unique step in the pathway. For C, well, there is only one step in that pathway. For A, then it's here. What do you think would happen if we block this one by one of these products? If we block this one, then it would mean we'd block everything else. So we might have enough A, but we might not have enough B, C, and D. If A was to act earlier in the pathway to switch off the enzyme controlling that, then that would be fine for A, but it would also mean that we would not have a high enough concentration of B, C, and D for whatever functions B, C, and D serve within the cell. Those ligands, we generally call them allosteric ligands, and the sites where they bind on the enzyme, we call them allosteric sites. We've seen this term before, and that simply means allosteric, that something foreign is binding to at a distant site or a distant place from the one we're interested in. So enzyme which converts S3 to A is means that it has an active site which can recognize and bind S3. A very commonly binds to another place on the enzyme, and this can give us changes happening within seconds. This example is a negative feedback, but it's possible also to have a stimulatory or positive feedback. But negative feedback is a much more common phenomena within our metabolic pathways. Other ways of looking at the effect of these allosteric effectors, which could be inhibitory or occasionally could be stimulatory. Sometimes people talk about them being competitive or non-competitive, similar to the discussion we saw of inhibitors in the previous section. But we usually, rather than that, refer to them as being K-series allosteric enzymes or V-series allosteric enzymes. So a K-series allosteric enzyme is one where the Michaelis-Menten constant Km is raised without having any effect on the Vmax, the maximum initial velocity that enzyme can function at, or at the highest substrate concentration. So a high Km, that means that the enzyme will be attenuated, its rate will be slowed down, because high Km means that you need more of the substrate in order to have the same reaction rate. So K-series allosteric enzymes can be made to be sensitive to inhibitors, which will make the enzyme less efficient and slow it down. The V-series allosteric enzymes work in a different way, or they're modulated in a different way. With them, Vmax, the maximum rate is lowered, but the Km remains the same. Now, the next section in Harper's biochemistry, which is the basis for these slides, considers the another class of allosteric effectors, which are called second messengers. And second messengers are a common secondary response to, for example, the binding of cytokines or hormones. So consider the hormone epinephrine that used to be called adrenaline. This can be bound by receptors on the surface of the cell. And there's a pathway acting via G proteins and the activation of adenylyl cyclase, forming cyclic AMP. Cyclic AMP is a small chemical which is an allosteric effector that activates an enzyme called protein kinase A, which is common to many cell signaling pathways. So we've got the first messenger, which is the hormone itself. The hormone, unlike steroid hormones, doesn't enter the cell. It binds its receptor at the cell surface and then mediates the production of a second messenger, another signal. And that second signal goes and activates protein kinase A. Calcium is another very common second messenger, which is found in the endoplasmic reticulum of most cells. And there's a receptor on the surface of the endoplasmic reticulum that is able to respond to signals and release calcium. So calcium is a second messenger. What else can affect enzyme functioning and enzyme activity? We now have the section on covalent modifications, and these could be reversible or irreversible. A very common theme is proteolysis, breaking proteins up, cutting them into pieces, and that's irreversible. You can't change that. Phosphorylation, on the other hand, which is something we've looked at before, that's reversible. Kinases add phosphate groups to serines and threonines and tyrosines, and there are specific kinases for specific amino acid targets within specific proteins. And the action of these kinases can be reversed by phosphatases, which remove that phosphate. So that's a covalent modification, which is very common for enzymes, and that's reversible. So irreversible and reversible. And we find the examples of these, and we'll look at some examples of both of these. The first example is chymotrypsin, which we've looked at before. And chymotrypsin has a catalytic triad of amino acids, that aspartate 102, histidine 57, and serine 195, which we described the mechanism of previously, and we call this a catalytic triad. But this catalytic triad is actually spread out over 3 pieces of the original chymotrypsin gene product, which goes through a series of proteolytic cleavages. First here between amino acids 15 and 16, then the removal of some more amino acids between 14 and 15 to lose this, and then we have another cleavage here at between 146 and 147, and between 148 and 149. So these very small peptides get removed, and we're now left with these 3 pieces, which are held together by disulfide bonds. And this then folds up into its active tertiary structure. The enzyme before the cleavage is called a proenzyme, and also it can be called a zymogen. So pro, the name in English speaks for itself. It's something that comes before. It's a progenitor of the active enzyme. In enzymology, people who study enzymes historically prefer this name, or have used this name, which is a zymogen. So a zymogen is the same as a proenzyme. A zymogen is something that will generate the final enzyme after a series of proteolytic cleavages. I say a series, in this case it's a series. It doesn't necessarily have to be a series. It may be, for some enzyme, maybe a single cleavage. Now this is not true for all enzymes, but there are a number of enzymes which are synthesized as a proenzyme. If we're interested in proteins that are not enzymes, or we want to talk more generally, then we call this the protein. In addition to the example of chymotrypsin, which is present in Harper's Biochemistry, I'll show you now another example is the coagulation cascade, which results in a stable clot based around the aggregation of fibrin after an initial deposition of platelets. In order to form this clot, which is essential for wound healing, we couldn't survive without that. The blood clotting can, of course, be a pathological, can be something which is out of control. So it's part of our normal essential physiology, as well as central to various pathologies as well. And a series of steps are required in order to build this clot from fibrin, and this requires that fibrin is converted from a pro-protein form to the mature protein. The mature protein indicated by the alpha, and the pro form, in this case, a white box, but importantly, there's no letter after it. And the amazing thing is that as we work back through these steps, in all these cases, what we're looking at is a proteolytic cleavage, which activates or converts a proenzyme into an active form of the enzyme. Factor 10A is factor 10 is the common joining point for 2 separate pathways, which lead into this common pathway, resulting in clotting. And in both of these alternative pathways, intrinsic and extrinsic pathways, also these blood clotting factors have to be converted by proteolytic cleavage into the active form. So there's a long chain of successive proteolytic cleavage that are required. And I won't answer the question, but let me ask it for you to think about, and that is, why do we need to have all these proteolytic cleavages? Why doesn't the cell just make the active form of the enzyme? You could think about this in terms of the blood clotting factors, and also you could think of this in terms of the previous example of chymotrypsin. What's the value to our cellular physiology of having enzymes made in a pro form, which is inactive, and require proteolytic cleavage to become active? And remember, this is for many enzymes, but this isn't for not for all enzymes. So Don't, because I'm focusing on it, don't assume that that's true of every enzyme. So those are reversible or irreversible covalent modifications. Well, clearly irreversible. We can't go back once we've and rejoin the pieces again. Now coming towards